Hi, Abyss. How are you? Thanks
Hey,
for
Kat.
coming.
I'm doing good. Thanks for the invite.
Hi, Victoria. How are you doing today?
Very well. Hello, friends. Nice to see you.
Hi, Victoria.
Hey, Abyss. Katarina. Hey, Zoila. Got a friend in the audience. Welcome. Welcome, Arif. I was reading the research and I was trying to imagine the little nematode worms aging more gracefully than they had previously.
Yeah, it's really interesting that plant. It The paper is really interesting because of the fat component. But I guess our speaker will talk all about it. I'm really looking forward to hear about it.
Yes, I'm pleased about that. Maybe help people relax about intake. Hey, Sissy Rahim.
Hey,
Hello.
How's your day going? Anyone else doing taxes?
no, you know what? I should do that soon. It's not going to be difficult though, because I just like submit everything to someone else to do it for me, but I haven't been able to do that.
That's a great method.
How long does it take? Mine doesn't take too long. I don't know. Maybe it's because, someone else does it, but
Well, I'm going on a few days over here. Hey, Jamie.
Hello, everybody.
hello and welcome, Dr. Bohnert.
Hi, thanks for having me.
Hi, thanks so much for coming.
Hello, Doctor. Thank you very much for coming.
Yeah, absolutely.
Hello.
Let's wait a few more minutes to let people have time to arrive, and then, if it's okay, I'll introduce you to the audience, give a little bit background information, and then Victoria asks like a more general question, like how you became a scientist type of question. I hope that's okay with you.
Sure, no problem.
Great, and after that, your the stage is yours, basically. Thank you. Everyone's so quiet. I'm sorry, I cannot unmute while I'm inviting people in. How is everyone's day?
I was looking up images of the Artemisia because I was thinking, Dr. Bohnert, if it could be fun for you to learn how to change your biopic there, which is really lovely with your initials. But if you didn't want a photo, you could screenshot something and then put that there in its place. So
Yeah, that's a good idea. I should look into that.
yeah, it's very fun. Like, for example, here, there. So now my PTR, do you see? Has it changed for you? Maybe it looks like something familiar.
Not yet. Not for me yet. But
If you pull the top of your screen down and then let it bounce back, we call that pull to refresh, then you should see
perfect.
a
Now
new
I see it.
image.
Yes.
I thought that would be more fun than being the nematode worm. Nothing
Oh, maybe I'll do
against.
that. How do I change my picture? I can do it right here.
Okay, so find a screenshot of an image that you would like to use,
Okay.
and then that'll be in your photos. And then once you've gotten the screenshot, should I tell you what to do next? Or should I wait?
Almost there. Give me 1
Okay.
second.
This is one of the best tutorials you can get for Clubhouse. And you can change it again.
Yeah, sorry for being.
You don't have to stick with this one.
Okay, I'm just gonna get
Oh,
a worm picture here.
true, Jamie. So,
Sorry.
Jamie, I am sort of, I guess it's not leafy. It's more, how do you describe the leaf form or the forms,
That would make
Dr. Bohn?
sense because my voiceover has been reading something that was leafy. I just assumed it was some kind of like, graphic for the room or something, but is that you?
Yeah, it's not, there's a different word that I can't recall, a botanical term for the shape of these leaf-like structures. But that's what it is. It's the Artemisia scoparia. That,
Yeah, Doctor, I
yeah.
was just I found an article about, I believe it's your work on Science Daily.
Okay.
Yeah, when you're like working on that big word for the name of the extract for the plant that I just don't remember. Every time I read it, the name's completely up my head as soon as it's done. But
Yeah, so it's
abs
right. It's the Artemisia scoparia plant.
absolutely, that looks absolutely fascinating. I had to like make myself to stop writing down questions because I was like, this person has a limited time. He can't move in. He can't just spend his whole time answering questions. But I'm very very excited for your talk. Yeah,
Oh,
because
great. Thank you.
So I'll let you know. I think you've probably found your screenshot by now, yes?
Yes.
Okay, so all you do is
Oh,
tap
I
on,
think I got it. Check if
okay,
it comes through now.
cool. Oh, okay, great.
Oh yeah, I see it.
Beautiful. Okay, so we've got a little nematode worm selfie in black and white. That's very dramatic, Doctor.
It looks very elegant somehow, I feel. I like
It's
it.
a great
It looks
image.
like,
It's a great visual.
yeah, it looks like a designer. See elegance.
Oh yeah, there's a lot of good ones I'm pulling up here. I wish I had a better one just available of even the fluorescent animals. Those are really
I'll try
beautiful.
to find them. What is their name?
Yeah, but just in one color. It's like, Karl
Which,
Lagerfeld says if you only dressed in one color, you're
Mhm.
always elegant. So, and the name is the elegant.
good
Anyways.
point. Good point. I'll look up the fluorescent in the meanwhile.
Okay, I think we can slowly start. So, welcome everyone to the Science Society. Thank you for coming, and a special thanks to Dr. Adam Bohnert. We are really honored to having you here today, and let me give you a little bit of background information about our guest speaker today. He's an assistant professor at the CDIB division, and he did his bachelor in science with summa cum laude in biology at the Rhodes College, Memphis, Tennessee, and his PhD in cell and developmental biology at the Vanderbilt University in Nashville, Tennessee. And he did his postdoctoral training with Dr. Cynthia Kenyon's laboratory, University of California, San Francisco, and Calico Life Science, San Francisco Bay Area. And his lab investigates the fundamental question in the field of aging, So, how are offspring formed from the cells of even very old parents born young? That question, I think it's really very interesting. And yeah, so he also received numerous awards, and from the W.M. Keck Foundation Medical Research Grant and AFAR Glenn Foundation Junior Faculty Grant, and so on. So yeah, it's a great honor to have you here. And Victoria, go ahead.
All right. Thank you, Katarina. So, Dr. Bohnert, Science Society welcomes you. And to make you feel welcome, it's really nice to ask a kind of a get to know you couple of questions. And so, my question for you is, when do you feel that you recognize that you had a particular interest in science? Maybe when you were a child or somewhat older in your life? And then how did that lead you to your current research?
Yeah, thank you all. I'm really excited to be here and to talk with you. So I think that's an excellent question, and when I'm thinking about it, I actually really don't know when I first um, got most interested in science. I had obviously been interested in biology when I was um, grade school and high school. And when I went to college, I always wanted to be a veterinarian. I've always had dogs. I love animals, and that's just it's what I thought I would do. Um, but then when I was in college, so as Katerina was saying, so I went to Rhodes College, which is a small liberal arts college in Tennessee. Um, and I did some research there, and it was mainly ecology based. Um, but then I had an opportunity to do summer research at Rutgers University in New Jersey. And when I was there, I was studying bacteria, trying to um, really specifically understand how bacteria survive in extreme environments, such as at deep sea volcanoes, because they have a really unique biology. And I just found the research experience, um, just sort of endlessly fascinating. That these new questions, there was always new excitement, and that everything was just different. Every day was different, every experiment was different, every question was different. And so that then, I think really inspired me to move on and do research. My PhD was in more cell and developmental biology using yeast to understand cell division, how cells physically divide from each other. Um, and then when I moved on to my postdoc, I became interested in this question of aging because it's a fundamental question of why we age, how we age, can we do anything about it? Um, and I thought some of the work that was being done was just really creative, and it got me interested, I think, to try to explore that field some. So yeah, so we're currently, so as Katerina said, we have a few different interests. We're interested in maybe natural ways to fight the aging process. How does nature do this? And one way that we look at this is by looking across generations, with this idea that if you look at humans, for instance, a human is going to have uh, a kid or a baby when they're maybe 30 years old, even 40 years old, um, and that baby is made from cells of that individual, but the baby's born brand new, right? So there has to be something natural about that. We're also interested, obviously with this work on Artemisia, we're interested in natural products that can possibly be applied to um, promote healthy aging, and so there's a lot of avenues that we're now getting at it. But yeah, I think it just started with first getting involved in research and really seeing the excitement of exploring just fundamental questions that I think really anyone can appreciate and be aware of. There are still mysteries, and I think there's a lot to be solved.
Thank you so much. That was really interesting. And I hear that you're something that, that's motivated you was not, well, the newness, the opportunity for always discovering something that
Yeah,
wasn't done before. And yeah, and this is what I feel like you're presenting to us right now. And we're all really excited to hear. So thank you.
yeah, absolutely. Yeah, it's sort of the newness of it, and really still the challenge of it. It's obviously a challenge to sort of pave something new, but being able to do really a creative pursuit, it's very fulfilling.
And with that, let's go into your presentation.
Okay, so today I can talk a little bit about the work that I believe Katerina has shared up here. It looks like the PDF file of this presentation, or the paper. So I just want to give a little bit of context for this, more so than what I've done already, and then I'll explain a couple of the experiments that we did as part of this paper, that show some of the main points. But again, one of the I think most compelling questions currently with aging biology research, um, is can you think of aging almost as a disease? Historically, in the past, there was some debate whether or not aging was a process that could be regulated, if it was something that just happened. Um, and now we certainly know that uh, it is controlled. It is under biological control. And that's something that we've really only started to grasp in the past 2 to 3 decades. Um, but sort of moving forward with that, it's how do you approach it? How do you, if you're going to therapeutically target aging, what does that mean? And so, I think one exciting entry point is this idea of natural products or natural product research, because there are several natural products that are known to act on biological pathways, um, that we can think of as being pro longevity or sort of pro health. And so that's a little bit of the inspiration for this project. So I've been an assistant professor at LSU, um, for about 4 years now. Um, and when I started, I was talking with some people, who work at what's called the Pennington Biomedical Research Center. And this is a research institute that's affiliated with our university, but they're very focused on metabolic disease, obesity, and diabetes. Um, and they have a center there. It's called a botanical center, where they have these natural plant extracts, and they've been studying them, several of these, for a while, because they think they have beneficial effects on metabolic health. Um, and when I was starting at LSU, and I was looking at some of the research that they did, I became interested in the possible connections with our research on aging, just because it's known that really this metabolic functioning, this metabolic resilience, is a core facet, that controls the rate or the pace, of aging. And so I was excited to think, maybe I could get uh, my hands on some of these extracts that they have available as part of this center, and to try some of these out using our favorite animal, which is the nematode C. elegans, um, which it lends itself really well to aging biology research because it has many of the same genes, uh, that you would see in mammals or even humans, uh, but it's only going to live about 2 to 3 weeks. So you can assess and analyze lifespan and aging, over a relatively short period.
Versus 30 years or so, as you were mentioning earlier.
Exactly, and so the really fascinating thing about this is that, um, when some of the original seminal work that was done by Cynthia Kenyon and others, identifying genetic mutations that that could make C. elegans live longer, dramatically longer, even double their lifespan. The obvious question was, how does this relate to, say, an animal that lives a couple of years or even decades, right? Does that relate at all? Um, and what's fascinating now is that others have studied, even in humans, centenarians, individuals that live 100 years or more, and what they found is that these same genetic changes that all these different groups of scientists have found can lengthen lifespan of C. elegans. These same genetic changes you detect just naturally in these long-lived humans, and so it really does suggest that these findings that we're making using these more tractable organisms, that they can give us real insight um, into possibly aging in a mammalian or a human context even. And uh, so when we first started this, there were a couple of extracts that we were working with, and we were mixing them in with the worm food on the worm plates, and we were putting the worms on there. And then what happens is you just track the worms each day. You come and you look at the plates, and by the time they're about 10 days old, they start to die off, and they'll continue dying off until they're about, again, 2 to 3 weeks old. Um, but you're looking at a whole population. You're looking at 200, 300 animals under any condition. So there's going to be some variation within that. And what was exciting was when we were putting them on one of the extracts, um, we were noticing that there were some differences. And if you can see the little picture that I took a screenshot of here and put as my profile pic, I guess, these nematodes, they're transparent, and so you can look at them and actually tell something about physiology um, just by looking through the microscope at the animals through bright light. It doesn't even have to be fluorescence. You can just look through bright light and you can see through the animal and actually see different tissues. And the actual first thing that we realized that was interesting was that the animals looked um, a little thicker. They looked a little thicker. They sort of looked fatty in a way. They appeared to be fatter than the control animals. Um, and one reason that was interesting was that this was how the extract that we were working with, that was how it was originally identified. So there was work done by Dr. Jackie Stevens, who's at Pennington, and they had screened uh, over, I think it was 400 botanical extracts to see if they could identify an extract that affected fat cell um, differentiation. And they found this one. And they found that if they gave this extract to mice that were on a high fat diet, that those mice were healthier, that they were metabolically healthier because of some action that this extract was helping with. Okay. And so this was interesting to us when we first started, because we thought, okay, well, we're giving them the worms this extract. At some level, it seems like it's having some effect. There's going being some response. Let's see what we see with longevity, with sort of aging. And so if you look at the manuscript, the paper file, the PDF, and you look at the first figure one, and you can look at the panel A is fine. Um, what this is this is a typical lifespan uh, plot or graph that you will see. And again, we're looking at days of adulthood on the bottom. So if you look at the normal worms, which are the wild type worms, WT control, that's going to be the black line. And the population's dying out really between 10 to 25 days or so. That's where that sort of line is plummeting down. However, the I guess more magenta and blue lines, these are going to be the populations that have been treated with what's labeled as SCO, which we call SCO, or SCOPA, which is for this Artemisia scoparia extract. And what you notice is that they're really living quite long. They're not actually starting to die off until after the other population, the control population, is maybe 50% dead by that point, right? And we did this in multiple replicates to try to understand just how much this was increasing lifespan. And so in B, this is demonstrating that. And really it averaged, or it hovered around 40%, a 40% increase in mean lifespan. And so this is fairly significant, and it's obviously robust. Um, and the other exciting thing about it that you see in panel C of figure 1 is that we also tried giving them this extract, the worms this extract, not really just throughout adulthood, but really starting at a later stage of adulthood. Because if you would think of, again, maybe some sort of therapy, the idea is maybe you're already giving this to adult animals that have already aged a little bit. And so when we did this in panel C of the first figure, in the lighter pink, what you'll see is that it still increases lifespan, um, about 20% in that case. Um, so not as much as when we started on the first day of adulthood, but it still is able to do it to a significant amount, right? And so that was something that was really exciting to us because um, we obviously thought, that may be a characteristic of some sort of, uh, let's say, dietary supplement that you would want to use as an anti-aging intervention. So, so once we got this and we really had, I think, the dosing down, the conditions down, and we had repeated this several times, we were pretty excited because obviously, again, the results were robust. They were very repeatable, and the animals were showing differences sort of by lifespan as well as just by eye. When we looked at them by eye, again, they appeared somewhat different. Okay, and so if you skip along, so we'll actually skip over figure 2. If you go to figure 3, which is right next to that. One thing that we were interested in looking at, again because of this history of studying this extract in more of mammalian cell culture, we were interested to try to see if it was affecting uh, fat regulation, if we could see any evidence of this, because we were curious whether or not, if it was changing fat, um, was that something that was going into this change in longevity. And so if you look at panel A of figure 3, what we're using here is it's a type of dye, a type of stain that's going to label fat in these worms. And so these are worms that are just laid out. And the idea is uh, the darker red that they appear, they're going to have more fat. And so the top 2 panels are the wild type, the normal worms. The very top one is just a control. And then the second to top one is the ones treated with this SCO or scopa, the extract. And what you can see is that the SCO treated worm shows more fat by this staining, and that's quantified in B. We also did some other assays to try to specifically pinpoint unsaturated fats, because there is, I think, emerging evidence that unsaturated fats are particularly powerful when it comes to sort of pro-health mechanisms. And so in panel C, we were quantifying fold change in unsaturated fatty acids, and what you'll see here is there was a significant increase with the extract. And then one other thing that was really, really striking was we decided to look at some of the enzymes that catalyze uh, the biosynthesis of these unsaturated fats or unsaturated fatty acids. And so we were particularly interested in 3 of these: fat 5, fat 6, and fat 7, which you'll see in D, E, F, G, and H of figure 3. And these are desaturase enzymes. They help to make the unsaturated fatty acids. And when we looked at the levels of these, so if you just look at panel E, this is maybe the easiest. We're looking at the protein level, so the actual levels of the enzyme, and we're looking by fluorescence because these desaturases are fused to a fluorescent protein. So they're fused to GFP here, so they're going to be labeled in these pictures as green. And so you'll see a variety of worms laid out in these images. And what you'll notice is on the bottom panels, you can actually see some green. Whereas on the top panels, which are the controls, the signal is much weaker, okay? And so for all three of these enzymes that help to make the unsaturated fatty acids, there's something about giving uh, the animals this extract that's turning on this program, right? You're making more of these enzymes. They can then help in the biosynthesis of the unsaturated fatty acids. Then, if we move on to figure 4, what I'll show you here is we wanted to test whether or not this uptick or increase in this pathway, right? This sort of fat regulation pathway, if this was causal in the lifespan extension, okay? And so what our idea was that we would take animals that did not have function of these enzymes. So again, FAT5, FAT6, and FAT7. And if they were really causal, this upregulation, this increase in them, if that was really causal in the lifespan extension, you would expect if you gave those animals the extract, they would no longer live longer, right? Because they wouldn't have the key components that are needed for that. And so if you look at figure 4, just the black line and the magenta line, these are just the wild type animals treated either with control or the extract. So again, you see about that 40% mean increase. And then I'll draw your attention to, I would guess they're sort of in the reddish pink colors towards the bottom on the labeling, where it says FAT5, FAT6, FAT7 RNAi control, and FAT5, FAT6, FAT7 RNAi SCO, okay? And these are going to be the lines that are furthest to the left on the plot there. And what you'll notice is that obviously these populations are quite short-lived. And also, when you give SCO, when you give the extract here, the animals no longer live longer. These curves are essentially superimposable. And so why this was, I think, really an important result was that we could say, or we could conclude that yes, it seems that these changes to fat, right, and in particular these enzymes that help to make unsaturated fat, that this induction or this activation of them upon treatment with this extract, that was something that was really important to increase the lifespan, extend the lifespan under those conditions. Okay, so it was giving a little bit insight into how it may be working. And then the last thing that I'll talk about is just figure 5, and I'll talk about this I think pretty simply and briefly. And the major point here is that there is a growing push in, I would say, aging biology research to focus not just on lifespan as a measure. And the reason is that, if you have a group of people, a group of 20 or 30 people, and you ask the room, who in the room wants to, if given the opportunity, would want to live longer, and you would be surprised that, I think a decent fraction of that group would actually say no. And I think the reason is that most people assume that if you're going to live longer, what that means is you're just going to prolong maybe that period of sickness or the period of decrepitude at the end of life, right? And so one important thing is that when you're studying sort of aging and lifespan, is that if there is a intervention that increases lifespan, it's really important to know if that's helping the animals to actually be healthier in later aging, right? Because you don't necessarily want an intervention that may increase or extend lifespan, but in the process, just extend the fraction of life where the animal is really unhealthy, because that would not be desirable. The obvious I think, goal is to make a scenario where you increase healthy years, right, at the end of life. And so we did some assays that were specific to C. elegans, that are measures of what we would call health span in a way, but really more health in later age here. And so what we've looked at in this is we're looking at stress resistance, and there is numerous lines of evidence from many organisms, not just C. elegans, but many long-lived organisms tend to also show a high degree of stress resistance, and indeed this can be considered really a marker of health. It is a validated marker of health in the worm. And the two that we're looking at here in terms of resistance, we're looking at resistance to a heat shock, and that's in panel A, and then resistance to an oxidative stressor, which is paraquat, which would be in panel E of figure 5. And so in panel A, what you'll notice is that if we just look at the black and magenta lines, that when subjected to a heat shock uh, in middle adulthood, so we'll say day 7 of adulthood here, the animals that had been treated with this extract up until this point, they survived that heat shock significantly longer. The thing that's interesting is if you look at the blue lines, so the solid blue line and the dashed blue line, these are the mutants that now lack some of these desaturase enzymes, the ones involved in the fat. And what happens when we do that is when you compare the two blue lines, now when you look at the animals that are treated with SCO, with the extract, the dashed line, they no longer do better. So again, it seems like this extract is promoting heat stress resistance, and it's doing so in part through some of these changes to fat regulation. If you look at panel E, it's going to show you something very similar. Here again, we're looking at survival after being given this oxidative stressor called paraquat. So again, the magenta line, the animals that are wild type that were treated with the extract, they're going to survive longer once given the stressor, but that improvement is now eliminated or removed when those desaturase enzymes, so in this case FAT6 and FAT7, when those are non-functional, right? So in that case, the SCO-treated version, which is the dashed blue line, that's actually doing worse than the control version in that case. Okay, so the take home with that is that this extract is not only helping the animals to live longer, but that we have some lines of evidence that these animals are actually healthier, at least in middle, trending towards later adulthood, which is encouraging. And the other thing that's interesting is both the lifespan extension and this improvement to stress resistance, it seems to require the changes to fat. It requires these desaturase enzymes that are helping to increase the levels of unsaturated fat in the animals treated with the extract. So I'll just sort of uh, summarize here at the end, and then we can talk a little bit if anyone has some questions. But there are, I think, 3 uh, interesting points here to think about. The first is that in this study, what we've found is that this extract is pro longevity, and that its pro longevity effects actually um, have something to do with the increased fat, and that may be something that is maybe counterintuitive or surprising to some people, um, because typically when we hear about fat sort of in a just general public discourse, it's often discussed as something that's bad, and here it's actually pro longevity. It's helping with stress resistance, and this fits in with a lot of emerging evidence. Again, suggesting complexities, right, and that there is this essential nature for fat, and especially specific types of fat, to be pro health, and in this case, pro longevity. There there have been, of course, several other studies that have been done to show this, including in the worms. So Ambrene's group at Stanford, they've done really beautiful work making this point. And in their case, what they've been looking at are genetic mutants that, again, are somehow dysfunctional in this regulation of the fat, and this compromises lifespan extension, right? In our case, what's a little bit different is we have this natural plant extract, which we can give the animals, and it almost turns on this program to mimic this response, right? And we're not actually changing anything at a genetic level. It's just giving this natural plant extract, and it's able to do this, which is, I think, really quite striking. The second thing is that one question we get is, are there any specific compounds or molecules that are known to be part of this extract that we could go after or potentially tackle as opposed to this complex mixture that's an extract? Is there really a defining compound? And so Dr. Jackie Stevens, again, who had done some of the initial work identifying and working with this scopa extract and fat cell differentiation. Her group has identified some bioactive compounds, but individually they don't fully recapitulate what the extract does. And this is a common theme, recurring theme that I think you see over and over again in botanicals research, where the extract really requires the complex mixture in order to bring about this effect. And so, as of now, we don't know. We don't know if there are any specific compounds that are, say, prevailing in this mechanism, but it would certainly be something interesting to explore a little bit more in the future. And then the final thing that I'll say is, again, C. elegans gives a lot of advantages for studying this type of problem as it relates to lifespan and aging, just because of the fact that it's relatively short-lived. Um, it would be exciting, especially given what's known about the effects of the extract on mammalian cell culture or on, say, mice fed a high-fat diet. uh, to further really pursue this or look at this to see the effects of the extract um, on mammalian, animal aging, especially now that we have, I think, some evidence from this invertebrate system that it can have a positive effect. And so I think if we found that, that would be something that would be, really quite interesting. It could also sort of direct new paths for studying potential interventions that maybe someday down the line, uh, we could move forward, making some sort of applications for humans potentially. But this is just a first step of getting at it and sort of defining it as something that potentially can play a role in some of these pathways, and it's doing so by what seems to be an interesting mechanism.
Yeah, thank you so much for this wonderful presentation and explaining this very interesting research, since I'm also aging. A question I have, like a more general one, because it was also mentioned that there are a few people that have higher body fat and are still, quite healthy and age quite healthy. And so does it matter basically where, like, if it's induced by this compound, or can it also, like, is there a general rule now for, like, you have less body fat, you live longer? Like, is there even a rule like this? Like, can we still?
Yeah, it's a great question. So I don't think there's a general rule, and I think there's complex regulation here, and that there are instances in humans and even in sort of experimental animals um, where you have individuals who are obese, but still very metabolically healthy under certain measures. And so the question then is, okay, well, there's fat in these different situations, right? But there could be variations in types of fat. There could be variations in where the fat is sort of held or stored, or where it's sort of located, right? Um, and also how that fat is mobilized, right? How is it sort of changing throughout the body? Um, and again, this is something that can be affected uh, possibly through diet, possibly through genetics. There are likely several branches that are working on this to influence what that is. Um, in our study, what we think is interesting, and I think again is mirrored in the more genetic analysis by the Brunet lab, is that if you have certain ways to increase this unsaturated fat, uh, whether you call it a stock here or a pool, um, that can be pro longevity, that can actually exert some sort of health benefit. Um, but yes, your question, I think, is something that is still, it's a it's not, I think, just very distinct, a yes or no. I think it's much more uh, context dependent. But it's really, I think, an interesting question for sure.
Yeah, thank you. I want to give everyone else opportunity to ask questions. So please flash your mic, and if you have a question, and then take your turn. So, okay, yeah, let's start with Abyss, Jake, Dr. Shah, Kyoko, and then Eli is coming to the stage. So, okay, go ahead. Thank you.
Yeah, I saw Jamie unflashing his mic, so I can probably go after him.
Oh, sorry, Jamie. I'm
Well,
sorry.
me, okay, me. Thank you. Thank you very much for the talk, Doctor. That was absolutely fascinating. One thing that's jumping into my mind right now, to take it to the beginning, the general thing. I was curious when you're working on, like you talk about, reversing aging and everything. Did you have any part of your investigation into this where you had to alter your perception of aging? Because of course, in literature and everything, we think of aging as like its own thing. Like, can I get 1 pound of aging, please? But it's not. It's a category describing a number of effects based on time, right? So when you
Mhm.
say aging, yeah.
Yeah, no, that's a great question. And so you're right. This is it's sort of a confounding thing, is how do you define biological age, beyond just the time aspect? How, if we're looking, say, at an animal or at a cell. So, obviously, again, one other thing that I think is really interesting with aging, um, from a biological standpoint, is if you are just walking down the street and you walk past a human, a person who is 60 years old, and a person who is, say, 20 or 30 years old, you can pick this out. If you walk past a dog who is 12 years old versus a dog that's 2 years old, you can pick this out. And the same thing actually applies for worms. If you're looking at a 15-day-old worm versus a 3-day-old worm, you can pick this out by eye. And so, a lot of what I do, so we didn't do it as much in this project, but a lot of what I do is actually at um, a cellular level, like really at the level of cells. And just like a full body of an animal, if you look at an individual cell that's making up a multicellular animal, um, there are certain hallmarks or characteristics that an old cell will show that a younger cell doesn't show. And so, going back to your question with this, how do you really define aging? At this point, it's really hard. We can, eh, from a cell biology perspective, from a molecular perspective, we can rely on some of these hallmarks, some of these appearances, some of these phenotypes, to sort of categorize a cell as looking old versus looking young. And we can also see how these change, right? And so for types of studies where we're looking at, you know, aging markers, you can sometimes there are natural instances where you see an aging marker sort of disappear or go away when given some sort of intervention. Okay, so again, these are something that you would naturally accumulate with age, but maybe you give an intervention, you give some treatment, or you look at some natural biological process, and these things naturally reverse. Now, whether or not that really means that age as a clock, as a time, as that is something that's being reversed, we don't know what's keeping that time, or really what that means. Um, but your point that there are several aspects of that are certainly true. We know this at the level of cells. It's not just one particular molecule or protein or factory within the cell, or DNA versus something else. These are all happening really at a systems level. And so, yeah, it's quite complicated. But there are a lot of people working on it, and there's a lot of, I think, really fascinating work that's coming out um, along the lines of cellular rejuvenation, right? How to at least at a characteristics level take a cell that shows signs of aging and to make it appear younger. And then if you couple that to some of these more physiological analysis, such as lifespan, or other sort of measures at more of really an organismal level, you can test if this is having some sort of effect, also, really at a performance of a full individual. Is that individual uh, maybe gaining more youthful health or characteristics?
Thank you very much, Doctor.
We had a guest speaker, but it's a while ago and I forgot his name. He's at Columbia University that did that developed a test in blood that looks at epigenetic markers where you can basically transiently look at aging markers. He
Right. There's a lot of research that I know of too that's looking at sort of epigenetic clocks. This is actually one aspect that we don't do as much of. We have looked at this somewhat transgenerationally across generations. We have a project that's related to that, but there are several groups that are really interested at the epigenome as part of this clock for sure.
Yeah, Dr. Shah, go ahead.
Obviously, it's your turn.
Thank you, Dr. Shah. Yeah. Hi, Adam. Great presentation, great paper. Thank you for walking us through each part of your findings. I do have a couple of questions. The first one being, did you notice any kind of behavioral changes in the two, like in the wild
Mhm.
type and the treated, um, like C. elegans nematodes? So the reason why I'm asking is that even though C. elegans don't have like this myelinated neuronal structure, that maybe the sort of like the delayed aging thing can actually manifest itself as the worms themselves actually becoming more active. Did you observe that? I'll start with that one.
Yeah, so that's a great question. And so we did test this, and this was actually one of the few changes that we did see. There were several that we weren't able to detect, but one thing we did see was that their movement changed, and this is something that's actually the opposite of maybe what would be considered a positive, right? And so what you're rightly suggesting is, maybe you would see that the animals are more active, or again, we would consider maybe a more youthfulness being that they're moving around and they're maintaining this movement capacity later into age. What we found with these animals was that they actually moved slower, and they did this from a young age. And so if we were looking at the extract treated animals, even from really the first couple days of adulthood, um, they were slower than the controls. Um, And from the way they were moving, it actually seemed like it was something where it's probably related to the changes in the worm body, that the worms were a little bit shorter and that they were just stockier. They were thicker, because I think, it's partly because of this fat that's accumulated. And so you're right. By this measure, it's something that's a little bit less appealing, um, because they aren't moving really as well when they're younger. But the thing that was interesting is, if you follow this out sort of with time as they're aging, um, they're going to get to a point where they sort of uh, they'll be at the same level as the controls, and then they'll certainly still be living longer than the controls. So after the controls have died, um, these animals are still moving around at that point. And so, yeah, this is something that we're still thinking about a little bit, and it's a great question. And we think there are other health parameters here that we've been looking at in terms of stress resistance. Obviously, with lifespan, and there are a few other things we've been considering. But this could come with some other effects or some other side effects. And in the worm, one of these seems to be, these worms are a little bit slower when they're younger, and that's probably related to the increased fat.
Gotcha. Thank you for that. My second question is, like, what is the actual mechanism as to how fat can actually prolong aging? Is it kind of alleviating the oxidative stress, or is it sort of acting like sort of an absorbent to, detrimental compounds that can be that may have been ingested because, some excess amount of like vitamins or any kind of opioids are fat soluble for the most part. So I'm just wondering if the fat is actually kind of reducing the distribution of these detrimental compounds throughout the body. Is that a fair way of saying how fats can actually reduce, or at least like increase lifespan?
Yeah, I think that's a fair possibility, or a fair sort of model to work with. Again, these sort of fat molecules, or lipids, um, they're going to be a normal part of cellular architecture, of cellular signaling. They're going to be involved in these different types of processes. And so you're right. There's a few different ways that it could work, and the specific mechanism that you're talking about here is certainly one that could feed into it. In terms of our um, our particular model, we don't know really downstream of the elevated fat, what is that necessarily doing. We know some of the upstream signaling, some of the upstream pathways that are helping to activate some of these things, these enzymes that are making the unsaturated fats. Um, but in our specific case, we haven't really pinpointed downstream of the fats uh, why that is. So that's a great open question, particularly for our experimental model.
So there was a paper, God, I think like 2015, that I believe was the first paper to demonstrate extension of lifespan, and incidentally also vitality, though I forget whether that extended to reproductive success or not, um, of antioxidants in an animal, but it was specifically combinations of antioxidants. If I remember correctly, it was vitamin E, N-acetylcysteine, and quercetin. Um, in rotifers, that's the important thing in rotifers. So what I was wondering is, are you looking to evaluate combinations, because it turned out that combinations were critical?
Yeah, so and that's sort of a key aspect of the extract itself, right? Is that by nature this extract is going to be um, a complex mixture of different compounds some of which, again, the Stevens lab has identified through fractionation studies by taking different fractions and trying to see what's there. We have yet to move on to the point of maybe taking some of these compounds and testing them in combination to see if, even if there isn't a single compound, maybe there are two, or some sort of minimal threshold there, some sort of minimal subset that would be able to do it. But yeah, you're right, and you can even think about, we think that this extract is working to extend lifespan by a somewhat specific mechanism, and that there are other longevity mechanisms that can naturally be tapped in the cell that are actually outside of this mechanism, that are sort of parallel strategies, right? And so you could even imagine giving this extract in combination uh, with something else that maybe acts by an opposite route, and seeing if there are even additive effects, right? If in combination they do even better. And that's something that is a really interesting question that we have not attempted yet, but it could certainly be done.
Dr. Shah, please
Yeah,
go.
thank you so
Is
much.
it my turn?
So, I don't know. I
Dr.
think,
Shaw, it's your, please,
sure.
please speak.
Okay. Thank you so much, Adam, for sharing your research. So, a couple of the points. First of all, it was very interesting to me in the first part because in the 1/3 of the lifespan, you keep track on delta 9 enzyme, and the second part was suggestion of the alternative with metformin from the extract that you just gathered from the C. elegans, and somehow with the possibility and of the gene cloning and transferring the genome to different animals, and that this is all possible right now, and there is a protocol for C. elegans specifically. This part was very interesting to me, but in the last part of the paper you mentioned about the bioactive compounds from ESCO which were identified, and specifically you talk about the cholinergic acid, which it was not enough that it can play a role or promote adipogenesis in mammals. So I was just wondering if you have any further information around this part and what other compounds, bioactive compounds, you found throughout your research.
Yeah,
Thank you.
so yeah, that's a great question. And so so currently we do not have any uh, further information on this. So there are some uh, novel bioactive compounds that are part of this, that are being studied by some other groups, and we've yet to again take sort of the compound approach to the worms, just because sort of I think the precedence of what's been found um, working in mammalian cell culture um, has yet to find anything that can really fully recapitulate um, the entire extract on its own. And so, yeah, I mean, this is sort of a question that we, as well as our collaborators that also work on this extract, um, that we get a lot is because typically, if you think so, you mentioned metformin, and that's a great example. And so metformin is, I think, very well known as sort of a diabetes drug. There are also some trials currently with metformin as a as a longevity intervention in humans. Um, and metformin has a an herbal, botanicals origin or sort of background. That's how that was originally discovered and described. But we're not at that stage yet with this extract. And one possibility may be that it's really just the extract itself which is the most powerful, because it's something about the complex mixture and the different things working together in some way. So the kind of unsatisfying answer, I think, is that we don't have any further information at this point. But if we could get to the point where we could test it and one way or another, I think get a better idea on the sufficiency of certain compounds versus the extract. I think that would really be a good step forward.
So your material was not transgenic, right? The C.
No.
elegans was not transgenic. That was wild species that you just used. I
Well,
remember
so
that.
it's a laboratory strain. So it's a laboratory wild type. It's called N2 strain. And so when we're doing just our standard feeding of the extract to these animals, it's just the wild type laboratory strain. So these are not wild isolates of C. elegans.
Okay, so thank you.
Wow, that's so interesting. I had a question about the botanical source of metformin, because as you had mentioned, sometimes the greatest power in these types of interventions is the synergy.
Oh yeah, yeah, certainly. So I'm not sure um, that I can speak to that as well. I'm not really the metformin expert, but I don't know. Could you just elaborate a little bit on what the question is, I guess?
Sure, yeah. I mean, well, as we know, a lot of different types of interventions have a botanical root source before they become synthetic and are
Uh-huh.
manufactured and distributed in that fashion. So I was curious, we're talking about C. elegans. Is there a like, where does metformin come from? But
Where does metformin come from in terms of the original botanical origin?
yes.
Oh, okay. Yeah, so I think there are herbal medicines that I think were originally traced back, I think, really to medieval Europe. So I forget exactly the name of what the specific herb was, but it was some sort of flowering herb. And these things are used sort of in historical or traditional medicine, right, where these different parts or different plants have a history. And this is the same thing with scopa. So this is really widely used in Asia, uh, almost as a diabetes treatment. Um, and so, yes, I'm not exactly clear on at what point metformin went from being more of an herbal origin to being more of, as you're saying, mass produced. In the terms of our studies, this is actually one of the limitations, is that some of the compounds that we are interested in working with from our extract, um, we don't really have sufficient levels of these at this point, or sufficient amounts of these to really um, get at that. And so that is certainly something with our progress as it relates to that, where we're currently limited.
Understood, yes. And here at Science Society, I'm not really sure is a valid answer.
Yeah, thank you. I think Jake, Kirko, and Shane, you were next. And I wanted to check with you, Adam, because we are a little bit over an hour. If you still have time for a few questions.
Oh yeah, well, it's fine.
Oh, great. Thank you so much. You've been so nice. Thanks.
Awesome. So my question is, is it possible for someone like myself to make this? If not, where do I get it? And is there any studies? And if there is, where do I sign up? Kind of
Yeah.
question.
Yeah, so this is one. So currently, so this is not commercially available in the United States. And obviously there's no approvals on this. Um, and so the simple answer is really no to most of those questions. The hope is obviously if we can push this research forward, there may be better evidence that this can be developed as a dietary supplement, or at least some aspect of it. But we're not really at that stage yet.
Bummer.
Might get to ask. Uh, I have a couple of questions. They're pretty quick though, I think. The first question, like I think you had said earlier about skull having a role in unsaturating fats. Am I correct when I say that?
Yes. Uh-huh.
Okay. So is there like the first question, is there any like preference? I don't know if you guys check for this for like the unsaturation to be cis or trans.
Yeah. So the only thing that we've looked at, so the the type of enzymes that we're looking at, they're specifically involved in making um, the monounsaturated fatty acids. So we haven't biochemically looked at monounsaturated fatty acids. We've looked some at unsaturated fatty acids, but not um, at monounsaturated fatty acids. But so you may have heard of these as these MUFAs or MUFAs. Um, but the fact that these three enzymes, the FAT5, FAT6, FAT7, so these are MUFA biosynthesis enzymes, and the fact that they're required, it really leads us to think that there's something about the MUFAs in particular. But there's more work we have to do there on that topic.
Sweet. And my second question is damn it, I think I forgot it. Wait, give me a second.
Yep.
Damn it, I just lost it again. Okay, I can't remember it. My bad.
That's fine if you remember. Just chime back in.
Hey Shane, go ahead. Welcome to the stage. Nice having you back.
Thank you. So my question goes a little bit ways back. I was curious if there were different results based upon the animals that the chemical was being tested on, as well as if there were any differences among humans.
So are you talking about in terms of like the genetic makeup of the animals?
Yes. My curiosity is kind of where those similarities are cross species. Is a question I was lingering in my head at a certain point when you were speaking, and so I don't know exactly where to cite that.
Okay. So let me try to answer it some, and just let me know if I can clarify any of the points. So um, the so we're using sort of a laboratory sort of wild type normal strain, and when we give them the extract, that sort of genetic background lives about 40% longer on average than that same wild type strain that's treated with control. It's just not treated with the extract. Um, there are certain though genetic mutants that when you give the extract to them, they no longer live as long. So some of these mutants are ones that have mutations in the genes that are for these desaturases, and these type of genes have um, what would be known as homologs or homologous genes, sort of similar genes in humans that exist. There's another gene that I didn't talk about, which is called DAF16. And what this is it's a type of protein that's called a transcription factor. And what happens is when we give the animals this go extract, uh, this particular DAF16 protein moves into the nucleus and it turns on all these different longevity genes. And what happens is when we look at animals that have a mutant DAF16, they also no longer live long when given the extract. And DAF16 is it's also known as FOXO in other animal species. And this is something that's known to be relevant to longevity in mammals too. So the way that this is acting, in principle, it could act on similar types of genes or pathways in human or human cell types. Um, just because of gene homology, and I think that the evidence that we have there is working with some of these mutants where we give them the extract and they no longer live as long. So we think that there's something causal about that. Does that help a little bit, or did you have a I guess addition to that?
No, that was great. Thank you.
I remember now.
Okay.
Oh, so my question is, are there any plans to maybe look into like metabolism? Cause like how I was kind of thinking about it was like if it's like has a role in making more unsaturated fats, would that then make uh the fats more available to be used? You know what I'm saying? Like even as it gets older, so like maybe it doesn't have to because it's like work as hard. You know what I'm saying? Like
Right.
the yeah.
Yeah. So one thing that's interesting with some of these um, some of the pathways that we think that this extract is working through. Um, if you look at mutants where these pathways are active, you see a similar sort of accumulation of these fat stocks. And you're right. It could be that they're just being held in almost a storage depot where they're not being mobilized, right? Or this lipolysis isn't happening, sort of the breakdown of the lipids. Um, and that it's maybe useful under certain scenarios. And so as towards really looking more at like a metabolic profile or a lipid profile, certainly, um, that would be something which, really, if that's not my lab's expertise, but it would be interesting to collaborate with another lab because it would be quite easy, in theory, to just take these different populations of worms and to see what's around, right? Even beyond lipids, right? Some of these metabolites, looking really at metabolomics, right? That's sort of I think a, a trending area of trying to understand the whole metabolome. So we don't know. Again, we actually don't know downstream of the fat um, in this particular pathway what it's doing, but yeah, I think your suggestion is a good one, and it could reveal almost unexpected results.
I had a quick question. So you had mentioned that you had stained for the saturated versus unsaturated fat. Could you tell us what technique you used for that?
Oh, so we didn't actually stain for saturated versus unsaturated fats. We were just using uh, there was a colorimetric assay that comes in a kit from a company where it detects relative levels of unsaturated fat. So it's not actually doing it relative uh, to the saturated fats.
Got it. And then you had mentioned that there's an overlap between the genome of C. elegans and hominids. Could you quantify that?
Yeah, so that's a good question. So I don't know exactly what the percentage is. Let me see if I can look it up really quick. But so C. elegans has about 20,000 genes. Okay, and
And a human?
the human it's not that much different. Okay, uh, give me 1 second. I'm just going to try to put in a quick search here.
I
Genome
believe it's
humans.
30,000 for humans
Yeah, I'm trying to find the
ish.
percentage. It says about 70 to 80% of the protein coding genes about similarity, about 83%, it seems like.
Is that substantially similar in terms of statistical analysis? Obviously, it's pretty cool that you can see right through them through bright field, right? Can't do that with
Yeah,
humans.
absolutely. And so the thing is too, when you're when we're looking at this and the genes that we're studying, so all of these different genes that we're studying, even just in the example of this paper, so this DAF16 FOXO, I didn't mention we studied this AMPK, which is a type of kinase. It's another type of enzyme. These different desaturases. We looked at different longevity mutants and supplemental materials, so it's like insulin signaling, TOR, which you may have heard of before, some mitochondrial mutants. These are all genes that have conserved homologs in humans as well as in these other experimental animals that people will use, such as like flies, um, mice. There are going to be homologs, right? One thing that could be different and that you do see a lot as you scale up. So, um, certainly when you move from something like yeast, so even like yeast is going to have many of these similar types of genes, and there is going to be overlap. But as you start moving to the human genome, with these different genes, you see many copies, several copies. There's going to be duplications. There are going to be genes that produce maybe different isoforms or really the same thing, but somewhat specialized, right? Maybe in a different tissue, maybe with a slightly different part. Um, and so, yes, it will build in complexity, but these core components, and especially the core regulation machinery that uh, the field has really defined to control the aging process. These are all things that you would certainly find as part of the human genome.
Fascinating. Thank you for that answer. And I was curious if cortisol was in part of the markers that you were measuring, given that there's overlap. And I can't imagine that, you had mentioned that the fat, you can tell the difference of between controls based on the assimilation of the fat or the production of fat tissue. And so I was curious, you know, is this a distribution that is even throughout the organism? Because in
Uh-huh.
humans, these types of manifestations tend to end up in the midsection due to
Yeah.
physics and all these other things.
Yeah, that's a great question. So we did not test cortisol specifically, but you're right. So when you look at by bright field at this, um, when you're looking at the extract treated animals, when they're on sort of a bright light background, the animals appear darker. So they actually they're not, they're maybe more opaque. Like they just appear darker through that. They almost look less transparent in a way. Um, and most of this fat. So if you're thinking of the worm, so the worm is about a millimeter, okay, in length. This is going to be the size of a period in print. Um, but if you so they're about a millimeter in length, and then you know obviously about the tenth of the size in the width. Um, Most of this fat, when we do this staining, so if you're going from sort of head to tail, you'll see it distributed sort of along this length. But a lot of it is going to be really in sort of the gut region, so then in the intestine, and that again is going to go along this whole the body axis, sort of from the head to the tail. So we will see that, and you will. So from the pictures that we have here that are really zoomed out, um, we're not making that out. One other thing that's a little bit of a complication here, that was something that we have been asked, is that um, when you look at just even worms as they are aging, their fat distribution, it changes. It's not as sort of localized as it was when they were younger. It becomes a little bit more disorganized and widespread. So this is a natural part of aging. And when we do our analyses, we're doing these not in young animals where the fat staining just actually looks beautiful. It's a little bit disorganized because we're looking at these at day 7, day 10 animals where the fat already just by this point uh, is a little bit different. But yes, the accumulation that we will see, it's it's not necessarily just in one part of the body. But I see your point. I see your point with sort of extending this, of thinking of it from a, a human and sort of maybe obesity or age-related obesity in humans, and where sort of fat tend to go.
It's completely different though, because this is a completely different medium. C. elegans, humans don't exist in soil or suspension. Our suspension is O2, right?
Yeah, yeah, yeah. No, you're completely right.
Thank you so much, Dr. Adam,
And
for sharing that. Yeah, I'll pass it on to Jamie. Jamie, you want to ask a couple questions? And thank you for spending so much time with us. We know we're already going over your time, but we just had a couple last questions that Jamie wanted to ask.
Sure.
absolutely, thank you very much, Doctor. This a couple of questions from an audience member, Marble. She wanted to know, she heard you use the word paraquat when you were giving your presentation, and she was wondering if that's what she thinks it is, being like an herbicide with toxic chemicals. Can you explain a bit more about why you would use something like that, like a toxic thing, in experiments like this with
Yeah, so
longevity?
that's exactly right. So that is true. You'll hear a paraquat talked of as part of herbicides, and part of how paraquat is working, or experimentally how we're using it here, is we're using it to induce stress. Okay, so that's exactly right. It is toxic, and if you give it at toxic levels, or at levels uh, where it can induce things like ROS, or reactive oxygen species, this is going to trigger some sort of stress response. Okay, and so this type of stress that is triggering, the first type of stress we were using in panel 5A, that was a heat stress. The paraquat stress, again, because it's this toxin that's going uh, to act as sort of an inducer of the reactive oxygen species, uh, this is going to cause oxidative stress. And so the reason that we're giving it is to try to see how well the animals can respond to this. If we stress the system out, um, are the animals able to better survive? Okay, and the idea, or what our finding was that even though we're giving the animals paraquat, and indeed they will die off. They're going to die off. Let's see, probably around 50, 60 hours after
The
the treatment.
botanical agent paraquat, just to be specific,
I'm sorry.
is paraquat is a fruit, or what is paraquat exactly?
Paraquat is just it's a chemical. I don't know what we're.
Sorry, paraquat is also, as far as I know, the common name of a fruit. So I was trying to differentiate that.
Oh, I'm sorry. Yeah, no. Yeah, and so if the panel E, so yes, so it is toxic. When we give it to it, the animals die off much sooner than they would have if we didn't give it to them. But all the animals in panel E were treated with paraquat. Um, but the thing that's, I guess, different about the controls and the scopa treated animals is that the scopa treated ones can still survive longer, just like they survive longer under a heat stress. So yes, they're dying off quickly than they would if we didn't have paraquat, but we give them a little bit of protection somehow if they've also had this extract.
Fascinating. And the last question she had was, do you have any thoughts on these balanced nature pills that she's heard that are around like the internet and stuff? Like, are they, like, what, are these just like junk, or?
I'm sorry, I didn't hear the middle part of the question.
She was asking, do you have any thoughts on balanced nature pills that she's seen around the internet, like advertised, and if they are like, nonsense, or if there's anything to something like that?
So some sort of like nature pills that are purported to have sort of longevity effects or anti-aging effects.
I believe
Is
so, yeah.
Yeah, yeah. So I don't know specifically specifically what specific pills maybe are being referenced in those scenarios. Um, and I think a lot of this from a pharmacological standpoint, sort of approaching aging from a pharmacological standpoint, is something that's very much up for debate. That is a new science, and that again, for some of these excellent questions that have been raised earlier in terms of how you define aging or how you define this clock. Um, is why it's still a very new field. And so what I would say is that, when it comes to sort of pharmacological interventions that are moving forward or progressing to trials to be tested to see if they have some sort of longevity capability in humans, there are some that you will hear about. So there'll be like metformin, as we've talked about. You may hear of NAD that also will come up when talking about sort of uh, metabolic functioning, metabolic health. There's rapamycin, which is a TOR inhibitor, and so the interesting thing with rapamycin is there's a study that's being done out of University of Washington where they're enlisting sort of just people as citizen scientists uh, to enlist their pet dogs in sort of a rapamycin trial with the idea that dogs live, maybe a decade or longer, but they're not going to live 80 years like a human. Um, so really from a pharmacological standpoint, that's where the science is. I think that's sort of where the consensus is. Um, Although there is a push to start, I think, viewing aging as a disease, and I think there will certainly be a lot of advancements from a drug standpoint. But it's going to take, I think, it's going to take a lot to sort of figure that out because again, some of our definitions are just they're kind of arbitrary in a way. And so and aging by nature, studying aging, studying longevity by nature, it just takes time. So I think time will tell.
Thank you very much, Doctor.
Yeah, thank you. I think that was all the questions that we had on stage. Um, do any of the moderators have anything else to add?
I have one question, Adam. By the way, you just talked about the DAP16 and not existing in a nucleus level, if I heard that right, correct?
I'm sorry, not existing in what?
So what you mentioned about the DAP16 and FOXO,
Uh-huh.
I might miss that
Okay.
part. I was
So
just wondering.
what we found is that when we give the worms the scopa extract, um, that the DAF16, which is the worm FOXO, that we see it redistribute into the nucleus of the cells in the intestine. And this is a typical um, relocation that happens when DAF16 FOXO is activated because it goes into the nucleus and it turns on all of these genes, many of which are longevity genes. And so we see this response um, when we give the scopa extract. And one thing that's interesting is that these fat, the desaturase genes, FAT5, FAT6, FAT7, these are actually targets of DAF16. So it makes sense that their expression, their levels would be upregulated in response to the treatment. And so what we've found with the mutants though is if you take a mutant which lacks a functional DAF16 and you now give it this extract, the animals no longer live long. They no longer live longer than controls. And that again sort of fits with this DAF16 being a core part of the response.
Under feeding stage or starvation?
So this is in fed stage. So they are fed. They're on. So the worm food is just this. It's an E. coli lawn, and so we maintain the animals during these aging experiments on this lawn.
Also with the growth factor, correct?
I'm sorry.
Also with, I mean, including growth factor, IGF or such a thing.
No, so it's just an auger. So it's a really simple auger that we have there that has just some standard salts in there, and then it's the E. coli lawn that's there. And the only thing that's different in our setup is that we've mixed in uh, the extract, which has been dissolved in DMSO.
Perfect, thank you.
Yeah, thank you so much. We've been going an hour and a half. I really appreciate all the time and um, sharing with us your amazing research. It's so interesting and so very helpful. So I hope in a few years when I need it, we'll have the
We hope in
compound
we
available.
hope in 100 years' time you can come back to the club and tell us how it all went right. Yeah.
Yeah, that's great. Well, thank you all so much for having me. I really enjoyed it.
Yeah,
Or
Thank you
maybe,
so much.
I was just going to say, maybe you could just package the worms, freeze dry the worms that you've used in your research, and then people can eat those, and it'll be an easy way to consume. Never mind, that's disgusting. Thank you so much, Doctor. This was really great having you here.
Sure, thank you.
yeah, and please come back anytime. For if you have updates on your research, if you would like to share more, you're always welcome back. And yeah, and also we have more guest speakers coming. So if you or anyone here would be interested in more guest speaker events like this, follow the club. And just to give you a short overview of the rest of the week, tomorrow morning at 9:30 a.m. we'll have Dr. Van Zunder. She is an amazing ALS researcher, and she made quite a breakthrough in recognizing a neuron toxin that is associated with ALS, and it's a potential new way of treating ALS. So I'm really looking forward to hearing her talk. And then on Friday, we have joining us from Israel, Alejandro Castrillon, and he will talk about ex utero mouse embryogenesis, so artificial utero work. I'm also really looking forward to that. And again, thank you so much, Adam. It was a great pleasure. And thank you, everyone, for asking questions. And yeah, have a good night, everyone. Thank
Yeah,
you.
thank you all. Bye.
Thank you.
Bye,
Good night.
everyone.
Good
Thank
night.
you.
Thank you.
Bye.
Good night, Doctor.
3, 2,
Bye
1.
bye, everybody.
Bye.
Bye, everybody.